All right, so hi everyone. This lecture is going to cover the first part of chapter 6, so genetic linkage and mapping in eukaryotes. And what we're going to talk about in this mini lecture is linkage a little bit, and then what the relationship between linkage and crossing over. So far we've discussed, in this course, we've discussed kind of what DNA is, how it works, and then we discussed what Mendel's discoveries meant for genetics and heredity. But we've kind of ignored something, which is the fact that genes, unknown to Mendel, of course, are present on DNA, and DNA is a physical entity, right? And when you think about DNA in your nuclei, you should be thinking of it as something that's present in chromosomes, right? And I've told you so far that, you know, the human genome, for example, contains about 23,000 genes, something like that. But you also know that the human genome only contains 23 pairs of chromosomes. So in order to fit all those genes onto those few chromosomes, then it's obvious, right, that each chromosome must carry many genes, hundreds or even thousands of different genes. And that's true. And so that gives us a bit of a dilemma when we're thinking about how Mendel's laws work. And indeed, it's true that if two genes are close to each other on a physical piece of DNA, on a chromosome, in other words, then that's going to mess up Mendel's law of independent assortment. So let's look at how the genome is organized in a little bit more detail. Basically, right, you have genes that are located on chromosomes. If two or more genes are on the same chromosome, they are physically linked, right, because they're on a long piece of DNA. So they are physically linked together. The term for that is synteny. So you say that two genes are synthetic if they are present on the same chromosome. They're physically linked by a DNA strand. Genetically speaking, if genes are close together on a piece of DNA, they tend to be transmitted from parent to offspring as a single unit, genetically. And those genes are said to be linked, genetically linked. Just to throw a spanner in the works, though, there's a phenomenon called crossing over, which is a phenomenon that occurs during meiosis I, where pieces of DNA are physically moved from one homologous chromosome to another one. They're basically swapped. That is crossing over, and we'll talk more about that in a minute. And that alters linkage. So it means that two genes can actually be separated, or two genes can be separated by crossing over. So let's examine some of these concepts and see what that means. So first of all, let's talk about linkage groups. Linkage groups are basically groups of genes that are all linked together. In other words, if you're a human being, you have 23 pairs of chromosomes. Therefore, you have many linkage groups because each chromosome is a separate linkage group, as shown by this carrier type down here, where we show all the human chromosomes, 1 through the X and the Y over here. And what you're looking at is essentially linkage groups. There's a bunch of genes on chromosome number 1. They're all linked together in one linkage group. Now, before we talk about exactly what crossing over does in terms of swapping DNA around, let's examine Mendel's laws of independent assortment and segregation one more time, real quick. So remember what we talked about previously is that as long as two genes or characters are on different chromosomes, they're inherited independently of one another in any combination. And two alleles, two copies of a gene, separate from each other during transmission from parent to offspring. That's Mendel's law of that's Mendel's law of segregation. Now, you guys all know how to apply those laws, because if I asked you what gametes this organism of the genotype big A, little a, big B, little b would make, you would all write down these 4 combinations, right? Because you could all do that. And hopefully you understand where those combinations come from the perspective of a cell going through meiosis. So Mendels Look here on the right is an example of these genes or this organism making its gametes, right? So here we have these different chromosomes lining up at the metaphase plate during meiosis 1. And what you see is that in this cell on the left, you've got big A lining up on the left, little a on the right, big B on the left, little b on the right. Remember that what this is showing is pairs of sister chromatids, right? There are 2 big A's here because this pair of sister chromatids represents 1 chromosome that has been duplicated during DNA synthesis, which is why there's 2 big A's. And the other chromosome is over here with the little a's. So this shows 1 possible combination. And when this cell goes through meiosis 2, eventually you'll end up with these A's and B's being segregated from these A's and B's to give you big A, big B, little b in the gametes. Mendel's law of independent assortment says that they could line up this way, but they also could line up this way, right? Big A on the left, little a on the right, same as before, but now we have little b on the left and big B on the right. And these would, of course, give you the other 2 types of gametes as this cell goes through gametogenesis, basically. And Mendel's law of independent assortment says that this is all random and all equal. So everything is equally likely. So 25% of the time, you're going to end up with this gamete, 25% of the time, you're going to end up with this one, and so on and so on and so on. So you get sort of, all 4 types of gametes equally split. So the question we're addressing today is basically, what if the A and B genes, as shown here, are on the same chromosome? So we're going to see a similar thing in the next slide, but instead of having 2 chromosomes here, A being on one and B being on the other, A and B are going to be on the same chromosome. So let's see what that looks like. So here's one example of an organism that is, again, big A, big B, little b. So we have big A here, little a here, big B here, little b here, right? So same genotype. But this time, A and B are physically linked in a linkage group on the same chromosome, right? So these guys could line up this way in meiosis 1, and then when they go through the end of meiosis 2, you're going to end up with these 2 sister chromatids being separated to give you these gametes, big A, big B. These 2 are going to be separated to give you little a and little b. Notice that what this actually gives you in the final count is 50% big A, big B, and 50% little a, little b. Now it's true that these could line up the opposite way around, right, according to Mendel's law of independent assortment, but the result will be the same. 50% big A, big B, 50% little a, little b. So this is in a case where there's no crossing over. These genes are tightly linked, and this is the result, right? So it's very different than what you saw before. These genes are segregating together all of the time because they are physically connected by DNA. So let's look at what happens to linkage when there is a crossover. So this example that I just showed you is what happens when there's no crossovers that occur. If a crossover is going to occur, it is, as I said earlier, literally swapping DNA from one homolog or from one homologous chromosome to the next. So again, remember what we're looking at here. This is the same situation that we just looked at. Genes A and B are linked on the same chromosome. what we're looking at here is the metaphase plate of meiosis 1. So these guys are lining up, ready to be separated. And what you see now is they're lining up as before, but this time we're going to see what happens if there was a crossover. Remember that this is one chromosome in red, the other chromosome is in blue. Sister chromatids are there because the chromosome has been replicated prior to meiosis taking place. Now imagine if the big B and the little b down here actually had crossed over during this process. So some DNA had moved from this version of this chromosome to this version of this chromosome. And there's two different alleles of gene B, right? So if a crossover occurs, we've physically moved this part of this chromosome over here, and we've swapped it with the big B. So you can see what's going on now. Here we have something different than what we saw previously, where this A, capital A and B, are going to form one gamete. Now, because there's been a crossover, we have big A and little b forming a gamete, and so on, which is now giving us 4 different types of gamete because of this crossing over event. So crossing over is one way that you can mix up genetic information from your chromosomes when you give them to your offspring. So what happens when you have a crossover is that you get gametes that look different from the parental cells, right? Which is really important because it means that when you pass on your genetic material to your progeny, you're not just passing on exactly the same stuff you got from your parents, right? There's some genetic mixing that's taking place. And that leads to some different terminology. So if you look at what we just talked about on the right here, when we had this crossover event, we now have these different types of gametes, some of which are the same as the parental cells, and some of which are different. And they're called different things. So these two in the middle are called recombinant or non-parental gametes. And they're called recombinant because a recombination has taken place. That's the name for when you switch DNA from one chromosome to another. And then non-parental because they don't look the same as the parental gametes. The other two on the outside here are called non-recombinant gametes because they were not affected by a crossover event, and they're identical to the parental chromosomes or the parental gametes. So when does crossing over occur? When does this mixing occur? This slide shows you meiosis, meiosis 1 being the top part, meiosis 2 being the bottom part. So the metaphase portion is right here. Crossing over actually occurs earlier during late prophase. So I'm going to zoom in on late prophase to show you this part in a little bit more detail. So what you see is this. If we zoom in on late prophase, we see that the two homologous chromosomes, one that you got from your mom and one that you got from your dad, are pairing up together because they have this, virtually the same sequence, and that causes them to pair together. Remember that this little X here, this little blue X represents 2 sister chromatids, right? This blue thing is one chromosome. It's been duplicated into 2 sister chromatids. And they're lining up together in what is called a bivalent or a tetrad, where you get these 2 pairs of sister chromatids lining up together. Now, what you see is when this occurs, sometimes there's synapsis of homologous chromatids and crossing over, which is what's occurring here. So this is where crossovers occur. You can see that in here, the DNA is coming to close proximity in between the 2 chromosomes, and 2 of those sister chromatids are exchanging blue and red DNA. So that's physically where that occurs during meiosis. So in summary, when you get crossing over, two genes that are on the same chromosome can look like they're independently assorting because they've actually switched chromosomes during the process of meiosis. Now, a two-factor cross studies linkage between two genes. So that's the example of genes A and B that we looked at earlier. Your textbook talks about three-factor crosses as well. We are not going to be concerned with three-factor crosses for this course. So don't worry about three-point linkage or three-factor crosses, just worry about two-factor crosses, which is what I'm going to continue to talk about today. So I'm going to give you another couple of examples of those just to kind of hammer the point home. This slide shows you what you were familiar with before this lecture, so independent assortment. So here we have two parents. This will be the P generation, homozygous. Each one is homozygous. This is homozygous dominant, two big A's, two big B's. This one is homozygous recessive. Genes A and B are on completely different chromosomes. Only one type of gamete can be made in each case, obviously. And when these guys were crossed, you get a dihybrid individual, big A, little b, big B, little b, right? If I was to ask you to write out what kind of gametes this guy would make if you crossed it to itself, what were the gametes of the F1? You would say, as we talked about earlier, every possible combination. And they would be in every possible combination in equal amounts, right? So 25% each. Big A, big B, little b, etc., etc. So that's what you already know. Notice that the parental gametes are big A and big B, and little a and little b appear. And you see 50% of the time you get identical versions of those, and 50% of the time you get non-parental gametes. And that's because everything is mixing as much as possible. Nothing is linked. Every possibility is going to occur at equal probability, so the numbers are all the same. Now, if two genes are completely linked, in other words, they're super close together on one chromosome, like these genes F and G, let's look at what happens in this case. So here we have a parent that is two big Fs and two big Gs, can only make one kind of gamete, big F, big G. Here we have a parent that is little f, little g, can only make one kind of gamete. And when they're crossed together, you get a dihybrid individual, big F, big G, little g. In this case, though, there's only one chromosome present, two homologous versions of the same chromosome. So if you were to write down gametes for this guy, there would only be two possibilities, right? Big F, big G, or little f, little g. Because no crossing over has occurred, they're exactly the same as the parents. So in this case, the parental gametes match them. So 50% come from one parent, 50% from the other parent. So that's no crossover, right? What if there is a crossover? So here we have, again, two genes, D and E, but this time they're on the same chromosome and they're far enough apart so they might be they might change by crossover occurring. Sorry. So crossover may occur this time between these two homologs. So this parent is two big Ds, two little E's. This parent is two little Ds and two big E's. Only one type of gamete can be made in each case, big D, little e, or little d, big E. And when these guys are crossed, there's this one possibility, a dihybrid individual, big D, big E, little e. But this time, there may be some crossover. So you could, for example, flip these two E's around from one chromosome to the other. If that occurs, you actually now get 4 different types of gametes. You get the original kind, big D, little d, big E, parental gametes. And you get ones where a crossover has occurred. And you can see how we've moved pink and purple DNA around here. In this case, these are the non-parental gametes because they're different from the parental gametes. These would be called non-recombinant types or parental types. These bottom 2 would be called recombinant types. One important thing I want you to think about or remember down here is that it is always true that the parental gametes are significantly more frequent than the non-parental gametes. And that's because crossover is a rare event, basically. So that's something to recall as we go forward, and I'll remind you of why that's important later on. So that's essentially all I have to say in this lecture. The other mini lecture is going to be concerned with the second half of the chapter, and we'll talk about mapping and how you do that. Okay, I will see you